Biology, Part 46. The external shape of the liver is of little importance as we talk about interlobular veins of the branches of hepatic portal veins, and the central vein of each lobule drains to the hepatic vein. Radiating from the central vein like the spokes of a wheel is a network of tiny blood spaces called sinusoids. Blood enters the sinusoids from the hepatic portal veins from the hepatic artery and slowly percolates through the central vein. The cells lining sinusoids are direct contact with the liquids part of the blood called blood plasma, and their surface area is increased by numerous microvilli. Between these cells fitted into the grooves, the membrane of the adjacent cells, there are tiny channels called bile canaliculi. Into the while the bile is secreted. There are no direct connections between the canaliculi and the sinusoids, so that the substance extracted from the blood must pass through the cytoplasm and the liver cells before entering the bile. The canaliculi from the interconnected network which drains to the bile ducts located in the interlobular connective tissues. Bile is produced continuously and is eventually collected and passed out for storage in gallbladder. The sinusoids also contain numerous Kupffer cells. These are vigorously phagocytic, engulfing worn out red blood cells, any bacteria present there. Functioning of liver. The functions of liver can be grouped under three main headings as follows. Digestive functions, then there are regulatory functions, of course, and the third one are the excretory functions. Digestive functions, regulatory functions, and excretory functions. We are going to discuss them all in detail. Let's talk about the digestive functions. Digestive functions of the liver are carried over by bile, as described earlier. Bile salts plays an essential role in the digestion of fats. In addition, sodium hydrogenobonate helps to neutralize the hydrochloric acid entering the duodenum from the stomach. The production and release of these substances is controlled by two main hormones, namely secretin and glycyrrhizin. Secretin is released into the bloodstream from endocrine cells in the small intestine, while acid is present in the CCK is released in response to the amino acid and fatty acids. Both hormones speed up the secretion and the hydrogenations ions into the bile, while CCK stimulates contraction of the gallbladder and causes the sphincter of the OD joining common bile duct to the duodenum and to relax. 95% of the bile salts released into the small intestine are reabsorbed into the body and can be reused. The liver synthesizes new bile salts to replace the 5% lost from the body in the facets. Regulatory functions. To keep them alive, all the cells in an organism body must be bathed in fluid medium. In vertebrates, this fluid, called tissue fluid, is derived from the blood, and its composition is very like that of the blood of plasma. The composition of plasma and the tissue fluid is kept constant so that the individual cells of the body are maintained in the ideal condition for growth and development. I'll tell you more about the same in my next video as I get along with them and give you the details.